Alright, gentlemen, now that we have finished Chapter 9, we are going to move on to Chapter 10, which is on meiosis and sexual life cycles. You'll find that a lot of the things we learned in Chapter 9 will translate kind of directly to Chapter 10, and we'll end this chapter and you'll end your studies this week by examining the differences and the similarities between mitosis and meiosis, because ultimately they are both types of cell division. They are ways for a cell to divide and create more cells with varying amounts of genetic information. So just to give some vocabulary again and remind you that we're finishing up mitosis and meiosis to move into genetics. So really what we're thinking about is we're thinking about cells dividing, is we're thinking about heredity, how traits transfer from one generation to the next. This is a small question in the bigger question of evolution that we've been talking about all year. And we know that variation is a consequence of this. If you guys look at your siblings, if you look at your aunts and uncles, you know, people who have the same parents don't necessarily look identical. Even if they are identical twins, there is still some variation between those individuals. And the study of all of this is genetics. So then the next set of chapters are going to let us study heredity and variation by looking at the genes that are there, figuring out why the genes have the results that they do. You guys have the benefit, as you've noticed, we skipped forward a few chapters and then stepped back. You have a lot of the molecular underpinnings under your control. You understand them in ways that the first geneticists like Mendel did not. So we kind of go in a reverse order here because now you understand what's happening at the small scale, and then we can think about what the big picture outcomes are. So the first concept of Chapter 10 is that offspring acquire genes from their parents by inheriting chromosomes. So you know, it's been known for a very, very long time that offspring look a lot like their parents, they act a lot like their parents, they have traits like their parents. It took until we understood truly that DNA was the genetic material to know that DNA, and ultimately DNA in the structure of chromosomes, is how offspring acquire those genes. So genes are the small units of heredity, as we've talked about. They're passed to the next generation in those cells that we discussed in the last chapter called gametes. So gametes are sperm and egg. So sperm and egg carry the genes from the parent to combine to make the child, the offspring. DNA is packaged into chromosomes, as we've talked about, and humans would have 46 chromosomes. Every organism is going to have a different number of chromosomes, but we'll do a lot of the math in this chapter thinking of the 46 chromosomes that humans have in all of their somatic cells. Just to give you another genetics term that you probably have not heard yet, is this idea of a locus. So the locus is the position on a chromosome that a gene is found. So as we start talking about pairs of genes and you know, genes from your mom and genes from your dad, we would expect the gene to always be in the same position on the same chromosome. So it would be a specific locus on chromosome 1, and it would be in both chromosome 1s at that locus. And hopefully that term will make sense as we keep moving forward. So there are two types of reproduction that we'll think about. One is asexual reproduction. That's just when an organism splits and makes more offspring, doesn't make gametes. Really, asexual reproduction can be accomplished just through mitosis. The cell just makes a copy of everything and then divides so that it has two identicals. And we call that offspring clone because it's identical. They're identical and they're all from the same parent. We're more interested in Chapter 10 in sexual reproduction. Sexual reproduction is when you have two parents giving rise to offspring. So they're combining their gametes, they're combining genetic material to create an offspring that has a new collection of genetic markers and basically variation. So here are some organisms that do asexual reproduction, tiny protozoa like Hydra. And many trees and plants actually reproduce by asexual reproduction. You've probably seen this with plants or trees that kind of just spread. They don't necessarily make seeds. They just kind of put out roots, and then a new plant will grow. So knowing that we have information coming from the parents to the offspring, and they can do that through either sexual or asexual means, let's focus in more on sexual life cycles. So sexual life cycles are a combination of both fertilization and meiosis. So meiosis is going to be a way to reduce the number of chromosomes, and fertilization is going to be a way to increase the number of chromosomes. So if you repeatedly decrease, increase, decrease, increase, you should stay at the same amount of chromosomes throughout the lifetime of the organism, or throughout the generations of the organism. So to think about that in humans, humans have, like I said, 46 chromosomes, or we often think about it as 23 pairs of chromosomes. And we can see these 23 pairs of chromosomes in something called a karyotype. So a karyotype, I'll show you in the next slide, is an image of the cells. You'll probably remember that we have one hanging in our classroom, and I've pointed at it before. And what you see in a karyotype is that the chromosomes that match the chromosomes that are the same chromosome from the parents, which we call homologous chromosomes, homologous pairs, or homologs, are put together on a karyotype. So we can see here karyotype, and you can see there's chromosomes 1, chromosomes 2, chromosomes 3, 4, 5, etc. And this illustration is showing you that each one of these lines in a karyotype is actually two sister chromatids from metaphase, and they are similar. You can see the colorations are indicating that the loci up and down this are the same. The similar genes are located in similar places, but we would not say that these two chromosomes are identical. We would say they're homologous because they're almost the same, but not quite the same. If you look at this karyotype some more as we get down to chromosome 22, you'll notice that chromosome 23, quote unquote, are X and Y. These are sex chromosomes that help determine the sex of the organism. In this case, an X and a Y means that this is a karyotype of a male. So there are X and Y sex chromosomes. Females would have XX. Males would have one X and one Y. If you're not a sex chromosome, that one special pair, the rest are called autosomes. So we have 22 autosomes, one set of sex chromosomes to make a human cell diploid. And we talked about this last week, but diploid has two sets of chromosomes, and we sometimes notate that with a notation of 2N, where N is the number of unique chromosomes. So in humans there are 23, but we normally have 46. 2N, 2 times 23 is 46. So here's just another illustration of that. You can have chromosomes, pairs of homologous chromosomes, and on top of this, inside of these duplicated chromosomes, you have sister chromatids. So just like last week, the terminology keeps adding more chroma things. So remember that you've got chromatids, chromosomes, duplicated chromosomes, homologous chromosomes. Just be really careful with defining exactly what it is you're trying to talk about. So what happens in the human life cycle is that the normally diploid somatic cells go through the life cycle, but the sex cells, the testes or the ovaries, will help make gametes through meiosis, which we'll talk about in a few minutes. Those gametes half the number of chromosomes, so they become haploid, and they have a chromosome count of N. So the only types of cells that humans make by meiosis are these gametes, and those gametes' role is to fuse with each other, a sperm and an egg, in a process called fertilization. And if you imagine, the sperm has N chromosomes, the egg has N chromosomes, N plus N equals 2N, you're back at a diploid cell after fertilization. We call that fertilized egg a zygote. And the zygote is going to have a set of chromosomes from each parent, and from that point forward, the zygote will just do mitosis until it comes into a fully grown human, a fully grown adult. So here's an illustration of this. You see that egg and sperm combine to become diploid, a diploid zygote. that diploid zygote will grow throughout the lifetime of the human, and then at some point the human will either make eggs in its ovary or sperm in its testis, and through the process of meiosis will regenerate sperm and egg, and this process will continue. So this is the type of sexual life cycle that we see in humans, and this is pretty common in most animals, that the organism itself is diploid and its gametes are haploid. So there are three main types of sexual life cycles, and I don't want you to focus too much on these, but I want you to realize that there is some diversity in this. All organisms that reproduce sexually have to alternate between meiosis and fertilization, but some of them do it in different orders. Like I said, animals are gametes are the only haploids. The rest of the time the organism is diploid. There are some plants and algae that actually alternate generations. So fully grown plants that you see outside sometimes are haploid, sometimes are diploid, sometimes are polyploid. They have even more copies of the genome than we think. And then there are some fungi and protists that are actually haploid for their entire life, and they're only diploid during the moment in time that they do sexual reproduction to try to shake up their genetic material. So here again is the cycle that we'd see in animals. The organism is 2N, it makes gametes that are in. You combine the in gametes through fertilization and create a zygote that's 2N, and then create a new multicellular organism. Some of these plants, on the other hand, you can have a, let's start down here, a diploid multicellular plant. It's going to undergo through meiosis to make spores. Those haploid spores can actually grow into a fully grown plant. These haploid cells would just undergo mitosis during this time. And then at some point in the life cycle of that organism, it will fertilize, mix with another haploid organism to create a zygote that can go undergo mitosis to become a full-grown plant. So in this case, you see mitosis happening within both gametes and non-gametes, diploid cells. As far as we know in animals, there is no mitosis that would occur with a gamete. Mitosis is only going to occur with these diploid cells. And finally, some of these fungi and protozoa actually live their whole lives in meiosis. So in meiosis, they go through, after meiosis, as a haploid cell, they go through mitosis, they grow into organisms, they do asexual reproduction, all as a haploid cell, and then at some point, they will combine in fertilization to create a zygote, and this allows genetic shuffling to occur so that you get some variation in the population. It then immediately goes back through meiosis again so that it can be haploid again for its life cycle. In particular, the picture here is showing you Chlamydomonas, which I know a lot about. Chlamydomonas only undergo fertilization. They only make gametes if they're under a lot of stress. So it's a stress response to changes in the environment that might kill off the organism that compels it to essentially have sex. Otherwise, it's just fine splitting on its own. Alright, so I've talked a lot about meiosis, but concept 10.3 is where we're actually going to look at the process of meiosis and see how it works. So meiosis reduces the number of chromosome sets from diploid 2N to haploid N, and it's going to take place in two steps, meiosis 1 and meiosis 2. And hopefully as we look at these, you'll see that both of them look a lot like mitosis. There's just some slight differences that occur. Because we go through two sets of division, meiosis 1 and meiosis 2, we don't end up with two cells at the end. We end up with 4 cells at the end, which is, just like going through the process twice. So to give you the big overview before we look at the specific parts, if you think about this cell in interphase, just like any other cell that's going to divide, mitosis or meiosis, those chromosomes do have to duplicate during the S phase of interphase. Once they're duplicated, in mitosis we would have seen these sister chromatids separating. In meiosis we actually see the homologous chromosomes separating. So these two sisters stay together, these two sisters stay together for meiosis 1. At this point, at the end of meiosis 1, we actually have created 2 haploid cells, and then going through meiosis II separates the sister chromatids to create haploid cells that have unduplicated chromosomes. That's the whole process here for meiosis. It'll get more complicated when we add more chromosomes, but just as you glance through this, we simply do one set of cellular division where we separate the homologous chromosomes, and then another set where we separate the sister chromatids. And that's what it says here. First meiotic division separates chromosome pairs, homologous pairs. Second separates sister chromatids. Meiosis II actually looks pretty identical to mitosis. It's meiosis I that is a little bit different. And as the result of this, you're going to get 4 haploid cells. So here is a big picture, and then we'll zoom in on the little pictures. But you'll notice that within meiosis I, you're going to see names that look familiar: prophase, metaphase, anaphase, and telophase. Telophase and cytokinesis. So as you go through meiosis I, separating those homologous chromosomes, you basically get them out of the nucleus, you line them up in the center of the cell, and you pull them to opposite sides before dividing into 2 cells. As we go into meiosis II, you're going to see the same thing: prophase, metaphase, anaphase, telophase. Prophase is going to be getting those chromosomes sort of arranged on the microtubule spindles. Metaphase is going to be when they're in the middle of the metaphase plate. Anaphase will separate them, and then telophase and cytokinesis will generate 4 haploid cells at the very end. So meiosis I, the first thing we want to look at here is the difference in meiosis I from mitosis is that we're actually separating these homologous chromosomes. So what you'll notice if you look at the metaphase plate is that you don't see individual chromosomes lined up, you see pairs of chromosomes lined up. So you see 1 red and 1 blue chromosome representing chromosomes from the mom and from the dad. The other interesting thing that happens during meiosis I is that these homologous pairs actually interact with each other. They do something called crossing over, which we'll talk about in a few minutes. Crossing over forms these structures called chiasmata, where the red and the blue chromosomes actually crisscross with each other. And what they do is they get so tangled up that when they get pulled apart in anaphase, you'll notice that a little piece of blue stays attached to the red chromosome, and a little piece of red stays attached to the blue chromosome. We call these recombinant chromosomes because they've recombined through the process of crossing over, and this allows even more genetic diversity to occur in these cells. So the important things that are happening here in prophase I is that we see this crossing over, the forming of chiasmata, so that homologous recombination can occur between the homologous chromosomes. We line them up in metaphase I and then separate them so that we now create 2 cells, and each cell will have 1 copy of each chromosome. So you see here we start with 1, 2, 3, 4, 5, 6 chromosomes. Now each cell has 1, 2, 3, 1, 2, 3. We've made 2 haploid cells at the end of meiosis I. After you finish meiosis I, you'll go into meiosis II, and I think meiosis II is a lot easier to understand. These 3 chromosomes will line up in metaphase II at the metaphase plate. This now looks exactly like mitosis. You've got 1 sister chromatid, the other sister chromatid. These chromatids will separate to opposite sides, giving you 2 cells in this step that are single chromatids and haploid. Because you brought 2 cells into meiosis II, you'll do this twice, so you'll actually end up with 4 haploid daughter cells at the very end of this process. So let's look a little more closely at crossing over. So in crossing over, you see an exchange between non-sister chromatids. Sister chromatids will be identical. Non-sister chromatids will be homologous. So it's important that you think about where we talked about the locus of a gene, because the locus of a gene is the same. If non-sister chromatids exchange segments of DNA, they should be exchanging the same genes, but those same genes might have some mutations that have been acquired over time, so that you're getting offspring with all sorts of genetic variability. This happens during prophase I, when they align allele by allele, locus by locus. Each homologous pair has one or more X-shaped regions called that chiasmata, and on that chiasmata you form what's called the synaptonemal complex. So I don't need you to worry too much about the complexity of how crossing over occurs, but just know that these chromosomes get tangled up, and as they pull apart in anaphase, they're broken and reattached at matching points. This is a really critical part of genetic diversity in all populations. So here's just a diagram of it. You see that these structures will crisscross. You'll get a crossover event right here. When that crossover event occurs, you will tear these apart, and you'll end up with blue over here and red over there, and you'll get a nice mixing of the genetic material. So comparing mitosis and meiosis, and I've been doing this throughout this talk, but I think this is the type of place that you want to be at the end of this, and your chapter assessment this week is going to include mitosis because I think that it's important for you to be able to distinguish between the two. There are 3 events that are unique to meiosis, and we just talked about them. They all occur in meiosis 1. One of those things is synapsis and crossing over. So this whole aligning of homologous chromosomes and crossing over of genetic material only occurs in meiosis. The alignment of homologous pairs versus just chromosomes to the metaphase plate also only occurs in meiosis. This occurs in metaphase 1. And then separating of those homologs, pulling a pair of sister chromatids, a duplicated chromosome across during anaphase 1 also only occurs in meiosis during anaphase 1. Like I said, once you get to meiosis 2, meiosis 2 is going to look a lot like just straight mitosis. So this is a nice diagram in your book. I encourage you to spend some time examining. You can see in meiosis, the individual chromosomes align in the middle, and the sister chromatids immediately separate. You end up with 2 cells that have 2N. In meiosis, you align the pairs of chromosomes. You pull the pairs of chromosomes to each side for meiosis 1. That's going to create 2 haploid cells. Those haploid cells then undergo meiosis 2, which looks a lot like mitosis, to separate the sister chromatids. Now you'll notice that these are not all 4 identical or even 2 identical, because the crossing over has created some variation even within these quote-unquote duplicated chromosomes now. They're not identical sister chromatids anymore. I'm not going to read through all this, but this is more summary information in your book that will help you kind of compare mitosis to meiosis. So what is the final result of thinking about these processes of sexual reproduction and meiosis? It's that we generate a lot of genetic variation that we can then invoke, or we have been invoking all year, that contributes to evolution. So for most of the year, we've simply been invoking mutations as the source of genetic diversity. We've talked about how it takes a long time, and you expect for some nucleotides to change. When those nucleotides to change, occasionally it changes the function of a protein, which occasionally creates a beneficial adaptation, which would be selected for under natural selection. These different genes that are created, these mutations create different versions of the same gene. We'll call those alleles. So when you think about some simple trait that we might talk about in genetics, like eye color, the gene that codes for eye color would have different alleles. Those alleles might be a blue or a brown. We'll find out next chapter. That's actually way more complicated than that, but I think most of you probably have that as a starting point. Sexual reproduction kind of puts genetic variation into high speed because it takes the chance mutations and allows us to shuffle them in various combinations throughout a population, and then by shuffling them you actually create more opportunity for natural selection for some offspring to be more adapted to their environment, and consequently have offspring that continue to be more adapted. There are 3 mechanisms that we will think about for contributing to this genetic variation that are parts of sexual reproduction. And those are the independent assortment of chromosomes, crossing over, and then random fertilization. And these are all 3 mathematical arguments, but I think there are things that will resonate with you as to how they create a lot of variation. So beginning with independent assortment, the idea here is that at metaphase 1 of meiosis, those chromosomes orient randomly. So in some pictures we saw red on the left and blue on the right. In other pictures you saw blue on the left, red on the right. Those are randomly oriented. So it's not that all of the red, quote-unquote, chromosomes go to one direction and the blues go to the other. It's like a coin flip. They can go any direction. So if you think about 23 chromosomes, 23 pairs of chromosomes in humans, right off the bat there, those chromosomes, every time you go through meiosis, have a coin flip going left to right. So you have 2 to the 23 possible combinations of chromosomes that you can get simply from going through meiosis 1. That's more than 8 million combinations of chromosomes right there. That's pretty good for creating some genetic variability, but that would also suggest that you know, you're kind of limited with how much variability you get there, that you only have 8 million combinations of chromosomes that you could make. The next way that we add some variability is through that process of crossing over. So now you take the 23 different combinations, but we need to add on the idea that those chromosomes can recombine. They can mix up in any place, essentially, along the chromosome, creating a new combination that we've never seen before. So now you take that 8 million, and you're going to increase it by a lot. Meiosis, on average, has 1 to 3 crossover events per chromosome pair. So you're going to create new chromosomes that have never been seen before through the process of crossing over. So that creates even more genetic diversity. And there's a nice picture of that there. You see we've got now a blue, a red, a blue with red tips, and a red with blue tips. These are all going to be different genetic combinations. And then the final way we see some genetic diversity is through random fertilization. And you can think about this in 2 ways. One is that any sperm can fuse with any ovum. So especially if you think about the number of sperm that are released at any moment when they're trying to fertilize an egg, any one of those sperm is randomly able to fertilize an egg. If you think in a population level, especially in other organisms, random fertilization can occur by different organisms actually combining sperm and egg, or in some animals, you know, multiple partners, multiple sperm with multiple eggs, lots of eggs. There's lots of variation in this, but if you think of 2 gametes, if you simply think about 2 gametes each with 8.4 million chromosome combinations, any zygote is about 70 trillion diploid combinations. So even among siblings in a family with the same parents, there's a 1 in 70 trillion chance that you guys will have the exact same chromosomes, which is well over the size of the human population. Just to kind of finish this up, evolutionary significance of all of this variation is that, as we've talked about all year, natural selection will act on these variations. Organisms that are better adapted will do better. They will have more offspring. Those offspring will continue to usually show those same traits. If some of those offspring develop a combination of traits through mutation or any of the other variations that we've talked about, that's detrimental to its survival, it probably won't survive, meaning that the offspring that have the beneficial traits will be more likely to survive through sexual reproduction. Sexual reproduction is better able to adapt to changes in the environment because of the diversity of offspring that any set of parents will get. Asexual reproduction is also successful. It's less expensive than sexual reproduction. It doesn't take as much energy cost. However, the organisms are all identical, saving for mutations that are generated through the process of DNA replication. So those changes happen a lot slower. Consequently, organisms that go through asexual reproduction tend to produce a lot more offspring, so that they can be more responsive to to changes in the environment. Both sexual and asexual reproductions can be successful evolutionary strategies. They just kind of accomplish it in different ways because at the end of the day, everyone needs genetic variation to have advantages in evolution and natural selection. That was a very quick run-through of the material in Chapter 10. We will talk about it in class a little bit more. Hopefully it made sense to you as we go through it. Thanks.